Hello everyone, this is BioBrain, and for my first video I would like to dive deep down in the wonders of the cell cycle. Okay, so number one, let's first define the cell cycle. It is a cycle of growth, development and division. Cycle of growth, development and division. Every cell has to go through the cell cycle. Again, it's a cycle of growth, development and division. Okay, and you might be asking, what is development? Development is aging, growth, aging. This is how a person that is 80 years old looks different from a person that is 10 years old. Their cells go through the cell cycle. That's how they grow and develop. So there are three main phases of the cell cycle: interphase, mitotic phase, or also known as mitosis, and cytokinesis. Interphase, there's a thing called G1 or gap1. Gap1 is an abbreviation for G1. In this phase, rapid growth happens and replication of organelles. So what I mean by rapid growth, the cell grows bigger in a really fast way. And replication of organelles. Let's define replication. Replication is just like saying Control-C, Control-V, copy-paste of organelles. Organelles is the organs inside the cell, organelles. So rapid growth, replication of organelles happens in G1. Then S. So what S means? S means synthesis. Synthesis means producing. Okay, so in this phase, copy DNA happens, or DNA replication, the same thing. DNA or like chromosomes, chromatin, sister chromatids. So DNA, it replicates in the nucleus. And then G2, preparation for cell division. Okay, first let's say G2, it means gap2, and it prepares the cell for cell division. This means it checks, is the cell big enough? Does it have exactly the right organelles, exactly the right DNA? Everything needs to be perfect before moving on to mitosis and cytokinesis. So it prepares the cell for division. So a summarized way: G1, quick growth and replication of organelles. Synthesis or S, DNA replication. G2 or gap2, preparation for cell division. That's the interphase. So let's move on to mitosis or the mitotic phase. The contents of the nucleus divides, forming two identical nuclei. So this is the definition of mitosis. The contents of the nucleus divide, forming two identical nuclei. So anything or everything that's in the nucleus, it divides into two. So once again, the contents of the nucleus divides, forming two identical nuclei. Nuclei is the plural word for nucleus. So in this phase, there are four stages: prophase, metaphase, anaphase, and telophase. So prophase is like the beginning stage. Chromosomes condense. Definition of condense is it appears. So condense. The chromosomes condense, and the nuclear material. Any nuclear material is like the nucleus, nucleolus, nuclear envelope. All that, it disappears, and spindle fibers form. So once again, chromosomes condense. If you're asking what did they condense from, They condense from chromatin, which is the unwinded version of chromosomes. So chromosomes condense, nuclear material, as I said, nucleus, nucleolus, nuclear envelope, it all breaks down, and spindle fibers form. Spindle fibers spindle fibers play a huge role in the upcoming stages. So the second stage, metaphase. In metaphase, the chromosomes group up in the middle, the equator of the cell. Equator means the middle of the cell. It all lines up into a horizontal line, in a straight horizontal line. Okay. So metaphase, chromosomes meet up in the middle, the equator of the cell, and they form a horizontal straight line, and that's the metaphase. So moving on to the third phase, the anaphase. The anaphase is a really long and tricky phase, so focus. First, as you can see here, chromosomes or sister chromatids separate. And where do they separate? Where do they go? They go to the opposite poles of the cell. So this is the first pole and this is the second pole. These are opposite poles of the cell. So one sister chromatid goes here, the other one goes there. So if you're asking what are they pulling, like what the sister chromosomes are getting pulled by what? Spindle fibers. Spindle fibers attaches to the chromatid, the sister chromatids, and they get pulled apart. And as you can see, the cell lengthens because the spindle fibers are shortening. That's this effect causes the sister chromatids to get separated and the cell to lengthen. As you can see here, the cell is just normal sized. As you can see, it's really long. So telophase, the most important phase in my opinion. And I would like to state first, prophase is the opposite of telophase. Opposite. They oppose each other. And in this phase, there's two groups. The two groups of sister chromatids from the anaphase, they get separated, right? There are two groups. So these groups of chromosomes, a nuclear membrane surrounds them, forming two identical nuclei. The two identical nuclei get ready for the next stage, cytokinesis. I would like to define telophase first. So as I said previously, telophase is the opposite of prophase. This means prophase, the chromatin condenses into chromosomes, but no, here the chromosomes decondense to chromatin. And the chromatin gets surrounded by nuclear membrane forming two identical nuclei. So these two identical, so let's move on to cytokinesis. The two identical nuclei, they have a barrier of cytoplasm between them. This barrier of cytoplasm ensures that this is one cell, but in cytokinesis, it divides. The barrier divides. So two daughter cells come out of it. Cytoplasm. Okay, so in cytokinesis, cytoplasm divides into two daughter cells. So the two nuclei become two daughter cells. And that is the 3 stages of cell cycle. Also, the interphase takes the most time of the cell cycle. The cell goes through interphase the longest, then the mitosis, then cytokinesis. Okay, and here we have a graph of the sister chromatids or chromosome. This entire graph, this entire thing is called a chromosome. This is entirely a chromosome. And you see these lines, each line is a sister chromatid. So this is chromatid, and they're getting held by a centromere. The centromere is in the middle. each sister chromatid goes through here. And you're asking why are there two sister chromatids? Because each sister chromatid belongs to one parent. Okay, so we have, I'm just gonna break it down. We have the cell, and in the cell we have nucleus. In the nucleus we have chromosomes. In the chromosomes we have DNA. In the DNA we have gene. Okay, so you know what a cell is, you know, and you know what a nucleus is. You know, a chromosome is what contains the DNA. Chromosomes have DNA, and DNA has genes, genetics. For example, your mom has black hair and your dad has brown hair. You could come out with black hair or brown hair, or you could have a mixture of both. Like you have maybe really dark brown hair, because your mom has black hair and your dad has brown hair. So so like you take the genetics from them. You can't have blonde hair, because your mom has black hair, your dad has brown hair. So you come out with one of them or a mixture of both. You can't have something that is completely different. And I would like to note, so mito, it means to wrap a thread. And what I mean by this, because in the DNA it looks like a thread, because multiple genes, multiple DNA are coming out of it. So it kind of looks like a thread. And cis means process in Latin. This is Latin. Cis means process. So it's a process of wrapping threads, which has involvement in mitosis and the cell cycle. And another word for division, the division is cleavage. So another word for division is cleavage. So again, that's cell cycle. The cycle of growth, development, and division. 3 main phases: interphase, mitotic phase or the mitosis, and cytokinesis. In G1, rapid growth and replication of organelles happens. S, the copy or replication of DNA. G2, preparation for cell division. Mitosis, the contents of nucleus divides, forming 2 identical nuclei. Cytokinesis, this cytoplasm divides into 2 daughter cells. Phases of mitosis. Prophase, metaphase, anaphase, telophase. This graph is a chromosome. Both of them are sister chromatids. This is a centromere. Okay, so we have cell, nucleus, chromosome, DNA, the gene. Note, mito means wrap thread, cis means process, the division means cleavage. Okay, so thank you for watching, and next video will drop next week, and I hope you learned a lot from this. Good luck.